Welcome to the Science Experience, your source for the most current and impactful research and preprints in science. Before we dive in, please take a moment to like this video and subscribe. It really helps support the Science Experience and allows us to bring you more deep dives into cutting-edge research. Okay, let's unpack this. Today we're taking a deep dive into something genuinely captivating. It's at the intersection of biology and artificial intelligence. We're talking about a new foundation model called GREmLN. Now, if you've ever felt, overwhelmed by the sheer volume of biological data, or maybe wondered how AI can make sense of that intricate dance inside our cells, well, this deep dive is for you. We're talking about unlocking secrets about cell properties, understanding complex disease mechanisms, even predicting drug responses, all through the lens of this groundbreaking AI.
Yeah, what's really remarkable here is how the researchers are tackling a pretty fundamental challenge. It's about applying these powerful AI models like, transformers, which we hear so much about, applying them to biological data. See, standard AI models, they thrive on sequences, think words in a sentence, but the expression of genes inside a cell, it just isn't sequential. There's no natural order. So that immediately raises the question, right? How do you build an AI that understands something without that clear built-in order?
That's precisely the challenge. So our mission today is to explore how this new model, GREmLN, and that stands for Gene Regulatory Embedding-Based Large Neural Model, how it gets around this orderless problem. And it does it by leveraging the intricate existing relationships within our cells. We'll look at how it actually works, the kind of massive data it crunches, and why its performance is genuinely turning heads in the scientific community. So to really appreciate GREmLN's innovation, let's first maybe hammer home this fundamental hurdle it's designed to overcome. Like you said, we often hear about AI foundation models for language, where words follow a specific sequence. Order matters. But when we look at single-cell RNA data, which captures gene expression in individual cells, there's just no inherent sequence to those genes. It's kind of like having a bag of words without any sentence structure. All the pieces are there, but no map based on position.
Exactly. And snapping back, the bigger picture is that this lack of positional meaning, it's been a critical hurdle for traditional transformer models. They were designed, fundamentally, to rely on understanding where things are in a sequence to model dependencies. Imagine trying to read a book if all the words were just shuffled randomly on the page. That's sort of the challenge with raw gene data, previous attempts. Well, they often just tried to force some kind of artificial order onto the genes, which, let's be honest, isn't ideal for capturing the true biological relationships. It's a bit of a workaround.
Okay, this is where it gets really interesting. GREmLN's breakthrough is integrating gene regulatory networks, GRNs, directly into its attention mechanism. So instead of forcing an order, it uses the existing scientifically known biological relationships between genes. It uses the cell's own wiring diagram.
Right. GRNs are essentially the cell's molecular logic. That's a good way to put it. They describe how genes interact, how they regulate each other, turn each other on or off, and how these interactions mediate cell states and functions. It's complex stuff. So by encoding this graph structure directly, GREmLN provides biologically informed gene embeddings. It's about leveraging these inherent cellular dependencies, including, crucially, potential causal relationships. It knows how genes are connected, not just that they're expressed.
So let me see if I get this right. The model isn't just seeing individual gene levels as numbers. It's actually seeing how they're connected, how they influence each other within that network. That makes the learned representations incredibly rich and biologically meaningful, right? Like the AI is thinking with the cell's logic.
Precisely. It gets that context baked in from the start.
That's incredibly powerful. But how? How do they actually bake this biological insight into the AI? Is there a specific technique? The paper mentions something called graph signal processing. Sounds complex.
It is quite elegant, actually. So graph signal processing, how does that translate into AI? Well, they apply something called a diffusion kernel to what's known as the graph Laplacian. Think of the Laplacian as like the detailed map of the gene network. The diffusion kernel then uses this map to figure out how influence spreads or diffuses across the network. Not just direct neighbors, but longer range effects too. So when the AI's attention mechanism looks at a gene, this process transforms its query. It makes it aware of its neighbors and even distant relatives in the network.
Ah, okay, so that's the secret sauce. It biases the attention mechanism to reflect those real gene-gene relationships from the GRN. So if gene A regulates gene B, the model intrinsically knows that connection matters more. And what about handling the sheer scale? Single-cell data is massive.
Right, millions of cells. So for that, they use a clever mathematical trick involving Chebyshev polynomials. It lets them efficiently approximate these complex diffusion calculations, makes the whole thing scalable to huge graphs and, the thousands of genes we have. It's computationally smart.
Makes sense. You need that efficiency. Now, to train something like this, you need data. Lots of it. GREmLN was pre-trained on, get this, 11 million single-cell RNA-seq profiles. That covers 19,000 genes, 162 cell types, across loads of human tissues, heart, blood, brain, lung, all sourced from the CELLxGENE dataset. So a really broad view of human biology.
And the data prep is key here. They don't just dump raw data in. There's rigorous quality control, filtering noisy cells, normalizing expression values. And they do this clever thing where they quantize expression into 100 bins, makes computation faster without losing too much info. But crucially, they generate unique GRNs for each cell type. They use a well-established algorithm called ARACNe for this. It infers direct regulatory interactions based on how genes' expressions correlate. And these ARACNe networks aren't just guesses. They're biologically meaningful, validated over years in cancer research, drug sensitivity studies. So the biological context is solid.
Okay, the proof is in the pudding, right? How well did it actually perform? They compared GREmLN against other big names in the field, scGPT, Geneformer, CELLxGENE, head-to-head.
Yeah, and on data sets it hadn't seen during pre-training, which is important for testing generalization. The results, pretty compelling. GREmLN came out on top across different tasks. Both cell-level tasks, like cell type annotation basically, identifying the cell type accurately said, and gene-level tasks, like understanding the graph structure itself, predicting connections in the network. For instance, on a human immune cells dataset, GREmLN got the highest classification score, a macro F1 of 0.929. Beat all the others.
And here's an aha moment, I think.
Right.
Get this. GREmLN achieved these top results with way fewer parameters, only 10.3 million. That's like less than a third of most competitors, sometimes a tenth. It really shows the power of embedding that biological knowledge directly, doesn't it? Instead of just throwing more parameters at the problem. Smarter, not your bigger.
Absolutely. And it also showed strong zero-shot performance, meaning it could accurately identify cell types it had never seen before during training. That's huge for generalization to new biology. Plus, on that graph structure understanding, task-predicting network links, it consistently outperformed the others. Even on tricky data sets like cancer cells, where the networks are known to be kind of rewired and different, it shows it can handle complex, even diseased biological contexts. Really impressive generalizability.
Okay, so stepping back, why should you, our listener, care about this new AI model? Well, this deep dive gives you a shortcut, really, to understanding how cutting-edge AI is unlocking the mysteries of our own biology. It's moving beyond just pattern matching to actually grasping the underlying logic of how our cells work.
Exactly. And this raises the question, how can we apply this? Well, by providing this unified, interpretable framework, GREmLN improves our ability to understand disease mechanisms, like in cancer, how tumors behave, and, crucially, how cells respond to drugs. Think about personalized medicine. Could we predict drug responses better? Design drugs that subtly rewire cell logic back to health? It's about getting to that root biological logic. That could really speed things up.
Of course, no research is perfect, right? There are always challenges. The current GREmLN model uses a static graph. The network structure is fixed. And its performance depends on how good those input networks are. Garbage in, garbage out, potentially.
True, but what's fascinating is that these limitations, they point straight to exciting future work. You could model graph dynamics, how networks change, that's more realistic, or incorporate other interactions, protein, metabolic pathways, microRNAs, build even richer, multi-layered embeddings. There's so much more biological complexity you could potentially integrate into the AI.
So that's our deep dive into GREmLN. A potential game changer, I think, in understanding the incredibly complex world inside our cells. It's such a powerful example of bringing deep biological knowledge right into the AI's core architecture.
Yeah, the idea that you can get these highly accurate, generalizable insights, maybe even with less data or compute, just by giving the AI the right biological context, that's pretty profound. It suggests a shift in how we build AI for biology.
Indeed. And here's a final thought for you to mull over. If embedding gene networks helps AI understand cells so much better, what other biological maps like metabolic pathways, protein interactions, even the 3D structure of tissues are just waiting to be integrated? What discoveries could that unlock? The future here feels wide open. We hope you enjoyed this deep dive on the SANS Science Experience. If you did, please don't forget to like this video and subscribe to the channel. Your support really helps us continue bringing you the most current and impactful research in science. Thank you for joining the SANS Science Experience, and we'll see you next time.